So good morning, guys, or good afternoon, or whatever. So this is gonna be one of the first recorded lessons, the video lessons. So in these lessons, I'm gonna have slides that you can stop anytime and write them down. By the way, you have to write anything in the slides down to your notebooks, and I'm gonna explain the topic to you. So pause the video whenever you want. Just write the things on the slide. Wait for the slide to finish. I mean, wait for everything that's on the same slide to be finished. Write them down to your notebook. And yeah, listen to me, and you can listen. You can watch the video how many times you want. Anyway, but make sure you finish the video. So today, what we're gonna do, we're gonna carry on with the food tests. We done starch, we done glucose. Now we're gonna carry on with proteins and lipids. So we're still on food. Our topic is food and digestion. So let's start with the food tests. The first one, the test for protein. So many foods include protein, such as especially eggs, for example. They have a lot of protein, or meat. Proteins are really important to us, as you know from the previous lectures. Let's see how we can test for proteins. So the test for proteins is called actually a red test. This is due to the color that is formed after the test. So to carry out this test, you need a sample. For example, you take some egg whites. Okay, you place this sample in a test tube. It can be dried, it can be whatever you want to test. You're gonna test this sample for proteins. Okay, so you mix it with water to dilute it. Okay, so you mix it with, of course, distilled water or pure waters. This is because you don't want to complicate your results, okay, contaminate your sample. Then you mix it, you shake it, then you add equal volume of dilute potassium hydroxide solution. This equal volume is equal volume to the water in your sample. After that, you mix it again, then you add 2 drops of dilute copper sulfate solution. Okay? And then these two solutions are actually sometimes mixed together. They're sold as in a bottle, and it's called a burette test solution. So you can buy this solution to do a protein test on a sample. Okay? Anyhow, so after you add the copper sulfate solution, if a purple color develops, a purple lilac color develops, it means that there are proteins present in your sample. Okay? That's simple. So write these down, pause the video if you need. I'm not going to repeat this again. You should pause the video and write these down whenever you can. So carrying on to the test for lipids. Lipids, as you know, they are fats and oils. Fats and oils, as you know, they are not soluble in water, but they are soluble in alcohol. So fats and oils are insoluble in water, but will dissolve in ethanol, which is alcohol. Using this fact, we can test for the presence of lipids. Okay, so let's see how we can do this. You need a drop from the sample that you're going to be testing, and then you mix it with ethanol. You dissolve it in ethanol, so to say. Then, like I said, the test will be shaken to dissolve it. The solution is poured into another test tube filled with cold water. So you take the solution of the sample and the ethanol, you pour it into another test tube with cold water. After that, if a cloudy white layer forms on top of the water, it means our sample contains lipids. You can notice this actually when you're eating, for example, barbecue, or you're eating lamb chops, and you have a, for example, ice cold bottle of soda, and then you see on the top of the surface of the soda, you see a white cloudy layer. That's actually lipids from the meat you eat. So if during this test, if that white cloudy layer forms after you pour it into the cold water, it means it contains lipids. The sample contains lipids. And this white layer is formed by tiny droplets in suspension, is called an emulsion. Okay, this thing that is formed is called an emulsion. An emulsion forms when the ethanol solvent dissolves in water, leaving behind the previously dissolved lipid in it. So when you put it into the water, the ethanol actually dissolves in water, then the ethanol leaves the lipids, which actually suspend on the top. Okay? Anyhow, so we have a video today to watch. It includes actually all the tests we have done in this topic. So let's watch the video. Yes.
In this video, we're going to be looking at the tests we do for different biological molecules, sometimes just known as food tests. We're going to be investigating fruit juice, egg whites, starch, regular cooking oil, and we're also going to use distilled water as a control.
So we have lipids, we have something that includes glucose, we have something that has protein, we have something that has starch. Distilled water is there as a control. Distilled water doesn't have any of these. So we're gonna see if these tests are actually true. Distilled water should test negative for everything.
The principle behind this is basically to find out if those different samples contain biological molecules such as starch, protein, etc. In order to do that, we're going to need to use these.
One extra thing we haven't seen here is there's a vitamin C test, but it's interesting. It's not our topic, but it's there. But anyhow.
PIP, ethanol, Benedict's reagent, biuret, and also iodine.
So you can see the actual solutions we actually use for these tests. For example, the iodine solution, the biuret solution I told you is the mixed copper sulfate and potassium sulfate. So this is for the protein test. And you have the Benedict solution for glucose, you have the iodine, like I said, for the starch.
Let's start with the iodine test, which is the test for starch. First, take your iodine solution and add it dropwise to each of your samples. Notice that the iodine is a kind of orangey colour, and when we add it into our samples, that orangey colour remains.
So you remember the actual colour of iodine was yellow, so if the sample does not contain starch, it stays yellow. And see what happens when it has starch, and what happens to the colour.
Until we add it to the starch solution, and there's a clear blue-black change in colour.
Exactly, so that's what you expect.
We continue adding this to all of our samples, and amongst all of them, starch is the only one that shows a positive result.
So that is expected. Fruit juice doesn't have any starch, egg whites do not have starch. Starch solution of course have starch, then it turns blue-black. Oil doesn't have starch, water doesn't have starch. Okay?
So to summarise, we use iodine to test for the presence of starch. You add it dropwise to each of your samples, and it turns blue-black if starch is present. Next, let's look at the biuret test for the presence of proteins. Take your biuret solution and add it dropwise to each of the samples.
So this is actually the mixed potassium and copper sulfate solution. Potassium hydroxide and copper sulfate solution, the biuret solution. So you don't need to do them separately because it's already mixed. You just add it to your sample to see.
If we give it some time for the reaction to take place, we can see in the egg white it turns a sort of purplish violet colour, and that suggests that protein is present. So to summarise, use biuret solution to test for the presence of proteins, add it dropwise to your samples, and it'll turn purple or lilac colour if protein is present.
Like we expected.
The Benedict's test is the test for reducing sugars. Glucose is an example of a reducing sugar. First, you take your Benedict's reagent and you add it dropwise to each of your samples. It's a good idea to label each of your samples for this test because you're going to need to put them into a water bath and they might get mixed up. For your water bath, there's a number of ways to do it, but the easiest way is to just boil the kettle and fill up a beaker with hot water.
So glucose test, as we done earlier, they're using water bath made from a beaker and just boiled water.
Put each of your samples into the water bath and then wait for a while. It does take some time for a reaction to occur, but if we watch this speeded up, you can see that one of our samples is clearly turning a kind of yellowy orange colour, and this indicates the presence of a reducing sugar. When we put them back into the test,
reducing sugar, guys, as you remember, it's the glucose or sucrose, okay? So it's actually testing for glucose here.
tube rack, we can see the fruit juice is the only one that's actually reacted to this, suggesting that it contains reducing sugars. So, to summarise, we use Benedict.
So it doesn't actually turn really brick red, but it changes color. It goes from colorless to a little bit light blue to a little bit orange, which means it has some sugar, but it doesn't have much. If we used a higher if you use a solution with higher glucose content, you will see a more brick red color.
Reagent to test for the presence of reducing sugars. Add several drops to your samples and then put them into a water bath. Depending on how much reducing sugar is present, it'll first turn green, then yellow, then orange, and it could form a brick red precipitate if there is a lot of sugar present.
Like I said, so the one that they tested here, they turned yellow orange, so it has some sugar, but if it has a lot of sugar, it turns brick red.
The ethanol emulsion test is the test for the presence of fats. First, you need to measure out 2 centimeters cubed of ethanol and pour that into each of your samples. With each one, you need to shake it vigorously. After you've shaken your samples, add 2 centimeters cubed of water and then observe the changes. Here in our oil sample, you can see this sort of white cloudiness formed. This is what's known as the emulsion. To summarise, we use the ethanol emulsion test for the presence of fats. Add 2 centimeters cubed of ethanol and then shake, followed by 2 centimeters cubed of distilled water. A white emulsion forms if fats are present. We use DC
fats, lipids, oils, anyhow, if they're present, you get white emulsion, the positive result.
PIP or DC pip to test for the presence of vitamin C. Fill your test tubes up with DC pip without.
This is the solution they use for vitamin C test. It's not our topic, but I thought it was interesting, so let's watch this as well. And you can tell me comments. What do you think about this as well?
Put your samples present in them. Then add your samples into it drops at a time and shake as you're doing it. The DC pip might turn pinky purple first. This is because of the acidity and doesn't reflect vitamin C being present. Keep adding your sample and shaking. If vitamin C is present, you'll notice that your DC pip turns colourless.
So what's the difference in this test? That's why I actually put it here. You're not actually having a test tube filled with the sample, but you are adding your sample in a test tube with your indicator. Okay, your indicator here, the stuff that you used for the test, like iodine for example. Iodine is an indicator for starch, but DC pip here is an indicator for vitamin C. So your indicator is a test tube, and then you are adding your sample drop by drop into the test tube slowly, and if it goes colorless, it means it has vitamin C. How quick it goes colorless depends on how much vitamin C it has. Okay, so let's carry on, see the other results. The first one, as you remember, is the fruit juice.
Repeat this process for each of your samples. When we make a final comparison, we can see that the fruit juice is the only one that has turned the DC pip colourless. So, we use DC pip to test for the presence of vitamin C. We add our sample to DC pip while shaking it, and the DC pip will turn colourless if vitamin C is present.
Okay guys, so this is the video. This is the end of our video lecture. So there will be more like this. Hopefully you are safe and well. And let me know, actually, let me know your comments. How did it go for you? Like I said, you can pause this anytime. You have to write whatever is on the slides to your notebooks. It's just like a normal lecture, but you have more time actually, and you can listen to what I say over and over again. So hopefully, hope to see you soon, and some recommendation about the incidents we're having. So just guys, just stay at home. Um, don't go out unless it's absolutely necessary, and be safe. Okay. See you soon, hopefully, and thanks for watching.